I'd like to thank the organizers for the opportunity to present our work. I have no disclosures related to this presentation. A goal of our work is to address unmet needs for certain CFTR mutations. For the purposes of this talk, I'll focus on nonsense mutations. Approximately 10% of CF patients have mutations that are unresponsive to current modulator therapies, and in addition, some patients respond poorly to modulators, have contraindications, or cannot tolerate the medications. So what is the rationale for adenine base editors? If we look at all human genetic variants associated with disease, about 60% of these are point mutations. And of these point mutations, almost half of them could be corrected if we used an editor that converted AT base pairs to GC base pairs. In the most recent CFTR2 database, about 60% of the disease-associated variants are point mutations. And of all these point mutations, about half could be potentially corrected using an ABE. So our approach is to use ABE ribonucleoproteins, or RNP, to make these changes. And this approach offers a rapid onset of effect and transient activity of the editor. So the design of the base editors is from our collaborator, David Liu and colleagues. And here there is a catalytically inactive Cas9 linked to a deoxyadenosine deaminase. There is nickase activity, and it's guided to an appropriate PAM by the guide RNA. The steps associated with the editing involve nicking on the opposite strand of the deaminase, deamination of the A residue to an inosine, followed by repair of the nicked strand, which converts it to a C, followed by further repair with installation of a G residue where the A previously resided. Some considerations for approaching CFTR mutations with this is the availability of an appropriate protospacer adjacent motif, or PAM. The targeted A residue must fall within the base editing window, and it's also important to consider whether there might be other A's within the editing window which could be subject to bystander edits. And we have selected 2 mutations as candidates for proof of principle studies. The first is 553X, where the mutation introducing this A results in a stop codon. If this could be edited by the base editor and converted back to a C, it would restore the arginine codon. In the case of W1282X, the mutant A introduces a stop codon, and appropriate repair of this would convert this back to the tryptophan codon. So our approach for delivery is to use electroporation of the ABE RNPs to cell lines or primary cells, as outlined here, Cftr cells, which are compound heterozygous with 553X, human primary cells with 553X on one allele and G85E on the other, and W1282X heterozygous cells with a deletion mutation on the other allele. 3 weeks after culture, we looked at CFTR-dependent chloride channel activity in Using chambers. We also isolated genomic DNA and prepared libraries for next-gen sequencing. So here's results using the Cftr cell line with the 553X mutation. The short circuit current is on the y-axis, time on the x-axis. In red are mock-treated cells with no evidence of CFTR-dependent current. In blue are non-CF controls with forskolin and IBX-stimulated current that is blocked by the CFTR inhibitor GlyH, and in green shows the current response in ABE-treated cells. The results of several experiments are summarized on the right. Mock-treated cells have no significant current. We see restoration of current in the ABE-treated cells, and this is in comparison to the non-CF controls on the right for FNI and GlyH. This panel summarizes the results in primary cells for the 553X on the left and 1282X on the right. Again, we see significant restoration of current with the ABE intervention in both cases. So we next looked at the editing efficiencies based on next-gen sequencing of the region we targeted. So these graphs show the total sequencing reads with target AT base pairs converted to GC as a percentage. For the CUFI3 cells, you see in the mock-treated 50% have the GC pair at that site, and that's because this is the other allele, delta F508. Following ABE treatment, we saw 91% frequency of the desired product at that site. For the 553X primary cells, we observed 77% frequency of the desired products. And for the 1282X, the number was 69%. We also saw a 16% conversion of the bystander edit there, the A next to the target with R1283G introduced. So we were particularly interested in the allelic editing efficiency. And to do so, we used this equation here. So for CUFI3 cells, 82% allelic editing efficiency was attained. And in the primary cells, 54% for the 553X and 38% for 1282X. Another important question is whether there is guide RNA-dependent DNA editing. So we used a computational program, CRISPR, to predict guide RNA-dependent off-target editing sites, and then we sequenced the top 10 predicted off-target sites, as shown here. And from those data, this shows the 10 off-target sites we sequenced and evidence of base editing. And we only saw base editing at the target site. The same result was obtained with 553X and 1282X, where we saw the on-target editing and a lower frequency of this bystander edit next door. So the summary so far with this work is that we obtained molecular correction of the 2 stop codon mutations using ABE 710 RNPs. The editing efficiencies we attained were sufficient to restore CFTR anion transport, and there was no evidence of guide RNA-dependent DNA off-target editing using the methods that I explained. We're also very interested in the delivery of ribonucleoproteins to airway epithelia. And we previously published using amphiphilic peptides combined with CRISPR nucleases to attain editing in vitro in primary cultures of airway epithelia, targeting CFTR allele. We attained the indel frequency shown here. We also delivered protein cargoes with an NLS, a GFP NLS, to the surface epithelial cell types. In vivo, we used the ROSA 26 MTMG mice, combining the peptide with either Cas12a or Cas9 RNPs. And if editing occurs, the cells will be converted from a red color to a green color, and as shown here, we saw diffuse editing along the airways with 12 to 15% editing in the large and small airways of these animals. So one of our questions is whether edited cells persist in vivo, and our colleague David P. at Felden Therapeutics recently completed a study where they delivered the S10 peptide and MAD7 nuclease, which is similar to Cas12a, to the airways of AI9 mice. And in the AI9 mice, cleavage of the LOXP sites will turn on RFP expression. So the mice received 2 installations of the nuclease, and then at 1 week, 1 month, 3 months, or 6 months, lung tissues were harvested to look at the gene expression. And you can see here, here's a negative control lung. Following MAD7 editing, you see a number of positive cells in the airways, and this continues at 1 month, 3 months, and now 6 months following delivery. Another question is whether RNP delivery can be repeated in vivo. To do this, David and colleagues performed a prime boost experiment. So in this protocol, AI9 mice received various combinations of the shuttle peptide, the nuclease, with an RNP not targeting the LOXP sites. And then 21 days later, were boosted, and then a week later, received the appropriate nuclease targeting the LOXP sites with the shuttle peptide. So here's editing attained with no prime boost with the nuclease. Here's with prime boost with just the peptide alone. Prime boost with the nuclease alone. Red cells is outlined here. And here's prime boost with the S10 peptide and MAD7, showing similar degrees of editing. So it appears that you can re-administer the nuclease using this peptide-mediated delivery approach in vivo. We're also interested in whether iterative screening would allow us to improve peptide-mediated RNP delivery. So in these studies, we used air-liquid interface primary cultures of human airway epithelia to deliver candidate shuttle, to use candidate shuttle peptides and the Cas9 RNP or Cas12a RNP to these cells. So on the top, following NGS, we have the indel percentage for the Cas12 RNP. Each number across the bottom represents a different experiment. And you can see for the various peptides, we have improvement in the indel frequency in replicate experiments. And similarly on the bottom for the Cas9 RNP, iterative screening allowed us to identify peptides with improved delivery properties. We screened more than 100 peptides with 3 or more donors per peptide. So the next slide shows results for Cas9 RNP editing in human and mouse airway epithelial cells. So first in human cells, we delivered a Cas9 RNP targeting the Cftr gene. And you can see with various candidate peptides, with S10 in the middle here, the starting peptide we used, we've been able to improve the efficiency of editing, indel frequency with newer peptides. And working with the Small Animal Testing Center as part of the somatic cell genome editing program, we were able to have the 237 peptide independently tested for efficiency in the TD tomato mice, again, converting red to green. And here are results of airways with RNP delivery with the S10 peptide, and on the right, results using the 237 peptide. where quantitative morphometry showed that we met or exceeded the editing efficiency of the previous best peptide. And we've also had the opportunity to look at delivery of base editing RNPs, in this case, the ABE8E protein, targeting the beta-2 microglobulin gene in human airway epithelia grown at the air-liquid interface. And as shown here, with the peptides optimized in our screen, we were able to advance the editing. We were able to successfully deliver the base editor and achieve editing frequencies in the 10 to 20% range. We also tested the ABE80 RNPs in rhesus airway epithelia targeting the CCR5 gene. And in this case, we compared to mock, we achieved editing in the range of 5 to 8%. So in summary, preliminary in vivo studies demonstrate persistence of edited cells and the feasibility of RNP administration using amphiphilic peptides as a delivery approach. Additionally, iterative design modifications yield peptides with improved RNP delivery to human airway epithelia compared to the previous S10 peptide. And our preliminary in vitro studies show evidence of CRISPR nucleases and ABE8E RNP delivery using newer amphiphilic peptides. And for additional information on these studies, you can see presentations by Sumba Terrari from my lab, and also her two posters, 610 and 620. So some of our future directions were interested in translational strategies for delivery of the ABE cargoes. So we will continue to explore the amphiphilic peptides in our models and also investigate dual AAV intene-extene vectors. We would like to further validate optimized methods for delivery in human primary CF cells and in animal models. And an important question for us is whether we can achieve CFTR gene correction in target cells of interest, including airway cell progenitors. And I'd like to acknowledge folks in my lab, especially Satish, Sumba, Katarina, Chris, and Amber, my longtime collaborator, Pat Sin, with Ashley and Christian in his lab, Joe Zabner, and Phil and Ping, who make the precious primary cultures for our group, our collaborator David Liu at the Broad Institute with Greg Newby, Mark Belke at IDT and colleagues there. Importantly, David Gee and the crew at Felden Therapeutics, and our funding sources here. And here's where I hang out. And thank you very much for listening, and I'd be happy to answer questions.
Hello, I'm Phil Santangelo, and I'm a professor in the Department of Biomedical Engineering at Emory University in Georgia Tech. And today I'm going to talk a little bit about our work on next generation nebulizer formulations for mRNA-based therapeutics for the lung. So I do not have any relationships to disclose regarding this presentation. So where are we now with regard to mRNA delivery to the lung? So to date, administration of mRNA to the lung has been, at least in the academic world, predominantly performed via intratracheal administration. And the problem with that is that even though it's useful and can certainly be used to demonstrate efficacy of particular mRNA-encoded proteins, I would say that overall the expression is not overwhelmingly fantastic. In addition to that, there could be very high animal-to-animal variability. There have been a number of polymeric vehicles, though, that have been that have been published. Many, though, are focused on PEI, and one of the issues with PEI is that it's quite clear that the toxicity of PEI, at least the therapeutic doses, could be an issue, at least regarding their translation into the clinic. What's probably been more, I think, a bit more promising is PBAE-based formulations, so polybeta amino ester-based formulations. These have been successfully used by the Anderson lab to demonstrate protein expression via nebulization in mice with minimal toxicity. Prior, though, I would say in general, with regard to the Anderson lab's work, I mean, they did show predominantly expression from reporter constructs. So recently, my lab we basically took their PBAE-based polymer and applied it in vivo for the expression of Cas13a, along with the delivery of guide RNAs, as a means of essentially degrading both influenza and SARS-CoV-2 RNA in vivo. So we were able to show efficacy against both pathogens using that particular polymer. So in general, we would like to move forward from that polymer and see if we wanted to determine if we could make new ones. I think the other driving force behind our wanting to make new polymers and new delivery vehicles for the lung is really had a lot to do with the recent trial from Translate Bio regarding the expression of Cftr mRNA in the lungs of folks with CF. And in general, unfortunately, their formulation was not able to improve lung function. And we were thinking about why that might be. And we really thought it had a lot to do with possibly screening. That can you properly screen for new vehicles? Because I think that's an important aspect of coming up with any new mRNA-based formulation. So we did two things. The first is we developed a number of new reporter mRNAs. So a whole suite that allow you to detect protein production and screen different delivery vehicles based on your readout. So we're going to use certainly luminescence as a means of examining as our main readout, but just the same, we have other reporters that allow you to assess protein expression and delivery using a number of different readouts. Anyway, so we use a GPI anchored nanoluke reporter for this process. So in addition to that, we also made a device. And actually, I would say we made multiple devices that work with mice, hamsters, rats, and ferrets. And essentially what they do, these devices have a very low dead volume. So they allow you to use much lower amounts of RNA than other approaches. And FDA-approved nebulizers can be incorporated directly into the device. So what this allows you to do is screen using very small quantities of RNA, lots and lots of different formulations, and in vivo, and through a nebulizer. So this kind of combines all of those aspects. So we essentially took new reporters, screening platforms, and we combined this with combinatorial chemistry in conjunction with the FinLab at Georgia Tech to discover new polymers for mRNA delivery to the lung. And so we started with a polymer building block that looks similar to what was published from Dan Anderson's lab a few years ago. And we took all of the different parts and pieces and we varied them to some degree. By varying them, we were able to come up with a large array, about 200 polymers to test. Fortunately, these polymers can be then complex with cargo, making particles that are around 100 nanometer in size. They can be then nebulized. As I mentioned earlier, they're screened through nebulization, breathed in by the mice. There's a schematic of the delivery setup here, which gives you an idea of how the mice are sitting in the delivery setup. There's a nose cone between that they stick their heads in, and these animals are not anesthetized when they actually are breathing this. So they're not anesthetized at all. The delivery is basically in the region between the two animals by nebulization through a vibrating mesh nebulizer. They then breathe it in. We then isolate their lungs and image them using an IVIS. So one thing that you can see here is that even with just a few micrograms of RNA, you can actually measure the luminescence in their lungs, and you can see that the variability between, even with just two animals, is actually fairly low. We then we made over 200 new polymers, lots of new backbones. We then went ahead and screened those. And again, you find winners and losers in vivo. And that's exactly what this graph is showing here on the right, is that you can easily determine the differences between certainly the winners and the losers. So we came up with about 5 new polymers in particular that were interesting for us to take a look at. And that's what you're seeing here. 76 was kind of our winner polymer. There's no question about that. And you can see some differences, but some of the others were also quite effective in delivering RNA to the lung. We did compare, we did look in vivo at the mean RNA intensity. So how much RNA was there in a region within their lungs? This was assessed by RNA scope. And you can see that 76 actually did was really, again, the winner there. We looked at zeta potential also, and 76 was around 60, which is kind of ideal for these particles. So when we looked at all of these particles, we certainly saw that 76 was very much looked like a winner. We then went ahead and actually delivered a number of different cargoes with this. We delivered antibody-based cargoes, we delivered Cas13, and we do have nanoluciferase-based reporters for both a full length for an IgG and also for Cas13. And we were able to get dose responses actually in both BALB/C and DBAs, and 76 performed quite well actually, even at fairly low doses in both of these species. We do get slightly better delivery in the DBAs. So you can easily move this into a hamster, essentially by changing the size of the tubes. And we did look at, again, average radiance, and we do optimize how much liquid actually is going into the nebulizer for different species. But we were able to easily take 76 actually from one species to another, and that's what you're seeing here is expression in hamster lungs. From that point on, we were able to move to ferrets. So here are ferrets in a tube and a nose cone, and you can see expression again in ferrets using the same polymeric delivery approach. And we were able to go into a non-human primate also. This is in conjunction with Francois Villinger's lab at the University of Louisiana at Lafayette, the New Iberia Research Center. And this is a macaque using a pediatric mask and nebulizer. And we were able to get had good success there also. So we were able to see 76 did perform better than 38 and 164, which we weren't too surprised by, but we could easily get expression throughout their lungs. And the radiances were about where we expected them to be. We also did performed RNA scope again in their lungs to look for the RNA, and you can see RNA actually well distributed actually within the lungs of these animals. And so this is just a zoom in view of where the RNA is actually being distributed. So what was important for us to do at that point is that we were able to deliver with the same polymer into hamsters, into mice, and into ferrets using 0.3 mg per kg and into monkeys. And one of the things that you'll see, it's kind of interesting, is that when you go from mouse to hamster to ferret, the actual amount of expression actually goes up, not down, which is what we would have expected actually. And we have a couple of hypotheses as to why that is, which I won't get into too much at the moment, but likely this is being limited by tidal volume more than anything else. But we do see it scale. When we go into a monkey, if you compare, actually, I should say, the ferret to the monkey, actually, interestingly enough, at 24 hours, the ferret compares very well to the monkey because they're not really, these two animals are not limited by tidal volume in terms of how much they're breathing. So this was certainly an advantage for us. We did perform dose responses, actually using both an antibody and Cas13 in ferrets, and we easily see, again, a very nice increase with an expression as a function of dose. So we can deliver into all of these species and deliver multiple cargoes. And I think this was sort of the take-home message, is that when we looked across species, as I said before, we didn't, and the NHP was pretty close to what we were seeing in the ferret, a little bit higher than what we're seeing in hamsters too. And I think just as the animals are getting larger, their tidal volume is a bit higher, and they're able to take in more of that 0.3 mg per kg dose. So a couple of other points that we think are important is obviously preliminary histology and tox from this particular vehicle. So in a mouse, we specifically looked at 76 and compared that to just sodium acetate, which is what the solvent is. We did look at preliminary RNA scope, excuse me, not RNA scope, but nanostring data, essentially gene expression data across, I think, something between like 500 and 600 genes. And essentially we found about four genes or so that were regulated, that were either six genes, that were downregulated and significant, 3 that were downregulated and 3 that were upregulated, that were significant. So really a very small number of genes that were modulated in the face of delivery. So that's really good to see. In addition, we did some biodistribution studies to see, is the RNA getting anywhere, is it going anywhere else and expressing? And in the mouse, certainly the expression is pretty much in the lungs only. I mean, that's the only place that we really detected any signal. We did also perform histology when we were delivering both antibodies and delivering Cas13 into the lung, and we really didn't see, there was really no histological changes of note that were, that are probably worth really talking very much about. But certainly in this case, we didn't really see big differences, both with our nanoluke constructs, with Cas13 and our antibody-based constructs. We didn't see any pathology at all. So this was also a good sign. And this is in a ferret. This is in, in a rhesus macaque. And the other thing that we did within the rhesus macaques is that we looked specifically at blood work, both at 4 hours post-delivery and 24 hours base delivery. And to be honest, we really didn't see much. There was not really much of a change at all in terms of any of the major pro-inflammatory. I mean, there was no IL-6 being made at all, which I thought was actually kind of extraordinary. I was actually surprised by that. So overall, we were quite happy. TNF alpha, no changes there. That was also good to see too. So IL-1 beta, no changes there. So again, in terms of major pro-inflammatory cytokines, we were happy to see that. So I think overall we were quite pleased. So I want to talk about efficacy using polymer 76. So one of the more exciting experiments that we did is that, as I mentioned earlier, we were interested in Cas13. Cas13 is an activatable RNase. You can think of it as kind of fancy siRNA, but we deliver an mRNA that encodes for Cas13 in addition to a guide strand. We published the first use of Cas13 against flu and COVID in a recent paper in Nature Biotech from my group. But one of the things we wanted to do is that we had used in that particular work, we had used the polymer from Dan Anderson's lab, great polymer. But because of the fact that Cas13, in general, we get very good delivery with the polymer 76, we thought, we should be able to lower the dose. And so we brought the dose down to 50 micrograms and actually compared that to a 200 microgram dose using the Anderson polymer. And in addition to that, we compared all of basically any of these results to IP administration. This is antibody 2381 from Jim Crow's lab. I do want to acknowledge Jim for donating the antibody to the cause. And so, but the difference is about 20-fold in terms of the amount of antibody delivered. But one of the nice things, so the way this experiment was done is that we delivered the antibody, we actually delivered the CRISPR, and they were given about 20 hours pre-exposure to SARS-CoV-2 in the hamster model. They were then infected with about 1,000 PFUs. And then 5 days later, we tracked their weights every day. And then at 5 days later, we made comparisons in progress, essentially. And one of the things that we found was that our Cas13-based drug at the lowest dose, 50 micrograms, compared extremely well with IP antibody. So we had 20 times lower the dose. This was administered by nebulization as opposed to IP. And we were able to basically have the animals recover just as well as the IP antibody. And then when we compared, I would say percent knockdown, we were very competitive with IP antibody, again, with 20 times lower dose. So we did see a progression where certainly as we went to the P76 did a better job, certainly than a little bit better than the Anderson polymer, at a higher dose for the Anderson polymer, and then a much higher dose of antibody. So overall, we were very pleased by the results. So, and this is RNA scope for SARS-CoV-2 in the hamster model. So in this case, all of the white dots are virus. And that's exactly what you're seeing here, is that essentially you're seeing the RNA dots in the infected case or all over throughout the lung. But what you can see is in the deep lung, especially, we were able to radically decrease the amount of viral RNA using Cas13. And this is just a comparison in similar parts of the lung. And you can clearly see the difference. It's about 80 to 90% knockdown in this region. So one other last thing I wanted to mention is that I mentioned mRNA-encoded antibodies. And so previously, we've demonstrated that you can express antibodies in the lung and that we can keep them in the lung for long periods of time, depending on how we actually modify the antibody. And so we've tested antibodies against SARS-CoV-2 and against the variant, and we had antibodies that work extremely well. We express them such that they're on the surface of the cell, and you can see that here in the bottom. This is super resolution imaging of the antibodies, and you can see they actually coat the surface here. And we did some durability examples, and when we can modify the antibodies such that it's actually for at least a week. Really, we don't see much of a difference at all in terms of the amount of antibody in the lung. And we did do an efficacy challenge, and one of the things that we did see was that the antibodies worked extremely well. I mean, any of the mRNA-encoded antibodies really caused weight changes that were very, very good, and it basically scaled with the IC50 of the antibodies. And you can see in the RNA-scope data that the antibodies were very protective in terms of decreasing the amount of RNA compared to virus only. So mRNA-encoded antibodies in the lung really look quite good. I just want to thank, so the gist of it is that we can, we have a new formulation using polymer 76 that works extremely well. It's very good at delivering to multiple species and can deliver multiple cargoes, mRNA-encoded antibodies in addition to mRNA-encoded RNases. And I do want to thank the folks in my lab. I want to acknowledge the Finn Lab, Hogan Lab, LaFontaine Lab, and Billinger Lab with all of the experiments. I do want to acknowledge Jim Crow's lab at Vanderbilt for supplying us with an antibody too. Thank you very much.
Hello, my name is Marie Egan. I want to thank the organizers for inviting me to speak to you about peptide nucleic acids, nanoparticles, and cystic fibrosis. I really have nothing to disclose except my grant funding from the CF Foundation and NIH. I'm going to talk to you about a project that was started a number of years ago with two collaborators, Mark Salzman and Peter Glaser. Our collaborative effort really combines technologies of DNA repair and nanotechnology. The question that we asked is, can we use DNA nanoparticles to correct mutations in Cftr? This is a project that was spearheaded by Nicole McNear and Rachel Fields. It uses peptide nucleic acids that are triplex-forming oligonucleotides that are capable of coordinating genomic modification in a targeted manner with nanoparticles to deliver them. And this is a question that we really want to answer. So triplex-mediated gene editing, how does that happen? Well, PNAs, these synthetic compounds with the neutral polyamide backbone instead of the usual phosphodiester backbone we think of in DNA and RNA, can be designed and synthesized to help correct a mutation that you have identified in a specific gene. You design them to target an area that is very close to your mutation, and then by providing donor DNA fragments, you will allow for this triplex forming molecule to alter the helical structure of DNA, triggering the endogenous repair systems of your cell to repair that DNA, and through recombination we wind up with a gene editing. We of course deliver the PNA and DNA in a nanoparticle. We use a polymeric system. It allows for different polymers to be used. They can be mixed and matched. There are many of them. And these polymers can then be chosen depending on what your cargo is so that you can optimize delivery. You can also alter the nanoparticle by antibodies, cell-penetrating peptides, an array of other additions that can be used to really improve targeting. These kinds of nanoparticles have been used in many, many studies. This is just a cartoon to show that they've been used to deliver nucleic acids in rodents, non-human primates, and humans, using from clinical trials. And in our own work, just to give you the power of how these changes can affect delivery, in human airway epithelial cells, which is what these three panels represent. The blue are nuclei, the PLGA particle, a blended particle, and the blended particle we used with MPG. What you can see is PNA and DNA were delivered. The PNA was TAMR labeled, so we'd see this lovely red color. And when you have PNA, and then you see this pink area within the nucleus, it's really that we've delivered directly to the nucleus. So I think you can see that this can be a very powerful delivery system. Possible approaches for delivery for the DNA and DNA nanoparticles that we wanted to use for this CF project. Well, we deliver to the respiratory tract clearly, topically, nose, breathe it in, airway, or think about it systemically, right? Can we deliver this systemically and really get to all the tissues affected in CF? So we have models to look at both delivery approaches. And we also deliver to airway epithelial cells grown at ALI, both human and mouse, to really try to work out some of the kinks in the system. So topical delivery to human bronchial epithelial cells was our first step. And what we found is we were able to develop particles and deliver them to airway epithelial cells. And then we looked at chloride efflux restoration in these CF-affected cells. So normally a CF-affected cell, if you load it with a chloride-sensing dye, you put it in a chloride-containing solution, and then you switch to chloride-free solution with some stimulation for CFTR, the fluorescence you get in our efflux assay doesn't seem to change. It's pretty much a flat line. What we're looking for is a change in the slope when we move from the chloride-containing solution to the chloride-free solution. And here you can see the same types of cells after treatment with these nanoparticles. Suddenly we have a robust change in our efflux. And then a summary bar graph here shows untreated cells, which have very little in the way of efflux, blank-containing particles, little. When we had our PNA, we said a much bigger response. And if you actually dive down into those cells, what you can see is there are some cells that have a robust response and some cells that look just like their untreated CF counterparts. And really is about 7% of those cells that are showing a much more robust response, closer to what we see in human bronchial epithelial cells, which is represented here in black. When you send samples down to deep sequencing, what you find is about 9.2% of the alleles are corrected when we treat with our nanoparticles, suggesting that we are getting some editing. That was very exciting to us, and we moved on to an animal model. We developed similar PNA DNA nanoparticles that would correct the defect in a murine model of CF. We used an F508del model at the time. We treated topically to the nose because we had a good nasal phenotype. We could do nasal PD in these mice. We know the change in response of the nasal PD in a CF animal versus a wild type. And if you look here in panel A, this is a response to IBM X04 skulls. In a wild type animal, you can see this gray zone and these red stars show that you get good hyperpolarization here. In a CF animal, you don't. These are pre-treatment, open symbols, blue symbols are post-treatment. And so in our CF animals, if we treated them with a blank nanoparticle or a particle that had scrambled PNA in it, pre and post, there's no change in this lack of response that we would hope to see in PD if we had any correction. However, when we treat them with our PNA designed to correct the murine defects, and we use our nanoparticle that is a blended particle with a cell-penetrating peptide, what we see here is pre and post. A large number of the animals fall into the wild type arena, right? And so that would suggest we would definitely getting some significant effects. We also see that effect not only in the IBNX and forskolin, or the part of the NPD, but also in the amiloride response, which is reduced. And then if we harvest those tissues and we look at them in the Husing chamber, we see an increased response to IBMX and forskolin in the treated nasal epithelium, much closer to the wild type compared to the CF, and it's inhibitable with CFTR. If you then look at the lung tissue, you can see that there's not a significant change in histology, so we're not seeing inflammation or destruction, not an increase in cell count, in fact it's a decrease, and not an increase in cytokines, suggesting that we had a fairly robust response. And we actually had over 5% of cells in the nasal epithelium corrected. We went on to look at the supranuclear epithelial cells grown at ALI. So this is a much more robust model of airway epithelial cells compared to our chloride efflux, because we have mucus, right? They're mature. And it was interesting because when we took the same PNA and our particles, and so now they have to get through the mucus, we found that our response here in short circuit current was much more modest. It wasn't terribly impressive. So Anisha Gupta, who was the postdoc who worked on this project, really started to look at how could we improve our delivery, how can we improve our reagents. And she introduced chiral substitutions into the PNA, allowing for pre-organization, for enhanced binding to the DNA, and this is what we call it gamma PNA. And with their gamma PNA particles, so gamma PNA, DNA, and the same sort of nanoparticles, suddenly we had a much more robust response. So it was clear that there was something specific about the gamma PNA along with the blended particle that was now able to get through the mucus, get into the cell, and give us a more robust response. That response happens without increased damage to the DNA. You know, whenever you do editing, you worry about DNA strand breaks. This is something called the Comet Assay. You can see untreated cells here, cells that get irradiated. Suddenly, oh gosh, we have a lot of DNA strand breaks. We have a long comet tail. We don't see that in either a PNA or a gamma PNA cells. So suggesting that this is a more robust correction without off-target effects. We went on to look at topical delivery in a mirroring model. Here we harvest the cells of our CF animals. We grow them, expand them, grow them at air-liquid interface, and then again treat with PNA DNA topically, basolaterally, and look at short circuit current as well as gene correction. Just want to point out, here's our CF PNA DNA. Here's the gamma PNA DNA, and what you can see as far as short circuit current response to IBMX and forskolin, CF versus our treated monolayers versus wild type. We're getting an increase response with our gamma PNA DNA particles, and that increase in short circuit current also correlates with an increase in gene correction. So via ddPCR, we can also see we go from about a 12 or 15% increase to about a 20% increase. So again, we could correct alleles, and we can see that functional response. So summary is PNA DNA nanoparticles are used to correct mutations in CFTR. This intervention is associated with functional activity. Polymer blend really is going to vary between model systems for effective delivery, and the level of correction varies from human and mouse. It's much easier to correct a mouse than it is a human. And the topical delivery is not associated with long-lasting correction. The data I did not show you. We've done serial NPDs in our mice after we've corrected things, and about 4 weeks later we need to redose them in order to maintain this effect. So what about systemic delivery? What are the advantages? Many organs and tissues can be reached. The potential to reach these many different cell types. Perhaps you could reach progenitor cells, and you would have a replenishing supply of cells. You wouldn't have to to do this very often. You would need to fight the mucus. But there are disadvantages to systemic reactions. Unlikely that one size is going to fit all when it comes to particles. And you have much less control over where your therapeutic is actually going. So we have two systemic approaches: an adult model, which is actually being spearheaded by Alexandra Petrovsky-Despot. And she actually has a workshop presentation that I suggest you all hear. And then Adele Riccardi has done in utero work with both IV and intra-amniotic delivery. She has been mentored by David Stittleman, who is a pediatric surgeon who has really shown her how to do this very difficult technique, which is quite excellent. So from a systemic delivery standpoint, we get baseline data, NPD and RPD. Then we treat the animals. They usually get 3 to 4 doses, 2 days apart over a week's time. Then we wait a week, and post-treatment, we repeat NPD and RPD, and then we wind up euthanizing mice post-treatment, looking for Cftr activity and correction. This is just a snippet of data that we have. There's nasal epithelium, rectum, and distal colon. This is a change in short-circuit current, ivermectin forced colon. What you can see is our treated animals clearly have a more robust response in the nose, in the rectum. It's modest but there, and in the distal colon. And this also correlates with a change in NPD response to ivermectin forced colon in our treated animals, at least a portion of them, not everybody. Again, it's 2 animals, very robust response. 3 others, there's a response, but it's not nearly as robust. And then 2 that don't seem to have responded at all. Trying to understand that who responds and who doesn't is a high priority for us. So we're just going to spend a little time on in utero editing. This is something that you take a pregnant mouse. You day at E15.5, you can do IV injections. At 16.5, you can do intraamniotic injections. You allow the pups to be delivered, grow up, and then we can do functional analysis on them. Just as a little snippet, here we have gene-edited tissues. We have evidence that we can edit the pancreas, at least 10% of it at times. Gut is also see up to 6% in editing. We do see a modest amount in the lung at about 3%. So we can do multiple tissues, which is pretty exciting. And that's a one-time treatment. And then there's evidence of durable correction. If we look at, we have a subset of mice that we allow to age. And at 4 months of age, if we look at NPD, what we can see in untreated animals, again, we don't see the hyperpolarization here. In utero treated, 3 of the 4 seem to fall into the shaded area, which is our wild type range. And then here's a group of wild type animals to compare to. And then short circuit current data, if you look at the GI tract, and this is rectum 8 months after in utero treatment, and again, this is a single treatment, you can see a modest response that is different from untreated, but not nearly as robust as wild type. But again, gives us evidence that this may be durable up to 8 months, which is a long time for a mouse. So we have DNA, DNA nanoparticles that can be delivered intravenously to correct mutations. There's partial restoration of Cftr activity is achieved. Multiple tissues can be targeted and can see this response. Delivery in utero may be associated with a more robust level of correction, which makes sense, and the polymer needed for effective delivery varies on the model system. We're also working on nonsense mutations. We've developed DNA nanoparticles for nonsense mutations Q542X and W1282X. We've treated human cells with good results, and we are now working on mouse models with these mutations, and we need to thank Craig Hodges from Case Western Reserve, because he's the person who's developed these models and has shared them with us. Moving forward, we also hope to develop DNA nanoparticles that will target multiple mutations at once. I want to thank you for your attention. I want to thank my collaborators, lab mates, the amazing group of folks at Yale, our team, our research center, and all of our funding agencies. And I want to thank you.
My name is David Bedwell, and I'm going to give a talk today about small molecule approaches to suppress CFTR nonsense mutations. These are my presenter disclosures with my funding and consulting agreements. All right, so let's start out with a simplified cartoon of translation termination. So when a ribosome comes to a stop codon in the ribosomal A site, a ternary complex made up of the translation termination factors ERF1 and ERF3, and ERF3 is bound to GTP, enter the ribosomal A site. This leads to the hydrolysis of GTP by ERF3, a conformational change, and which leads to a combination of ERF1 into the PTC or peptidyl transferase center. It then promotes the release of the completed polypeptide chain or the nascent chain if it's at a premature termination codon. And then you end up with a post-termination complex. So the key players here are ERF1, ERF3, and a ribosome with a stop codon in the A site. All right, so now let's talk about nonsense mutations in genetic diseases. So there are, according to the National Organization for Rare Diseases, there's something like more than 6,000 rare genetic diseases. About 25 million people in the US have a rare genetic disease. So in-frame premature termination codons, or PTCs, account for about 11.5% of all described gene lesions, so all mutations that cause inherited human diseases. And about 3 million patients in the US carry a nonsense mutation that results in an in-frame PTC. So if you look then at CF, there's more than 30 people in the US living with cystic fibrosis and more than 70% worldwide, and about 10% of these patients have PTCs. So there's a significant number of people that could benefit from nonsense suppression approaches. All right, so here's what happens when a gene has a nonsense mutation. First of all, the primary transcript is made in the nucleus. It is then capped, spliced, the 3' end of the message is cleaved and polyadenylated, and then it gets exported out of the nucleus into the cytoplasm. In a process called the pioneer round of translation, the initial ribosomes bind and translate this transcript. And that's shown down here in more of a steady state configuration. And if there's a PTC, you basically translate the first half of the mRNA and then terminate prematurely, so resulting in a truncated protein. So there are two major consequences of having a nonsense mutation that leads to a premature termination codon. The first is synthesis of a truncated protein. And then as a secondary consequence, this leads to destabilization of mRNA by nonsense-mediated mRNA decay. So it basically reduces the steady state level of the mRNA, denoted here by the dashed line. And so this can be down 2-fold to 20-fold. And so this can have significant consequences on the ability to express these mRNAs. So what we would like to do is find small molecules that can suppress this premature termination process. And so when you have this premature termination codon, you terminate generally quite efficiently, so let's say 99.9%, but it is in equilibrium with the insertion of a near cognate amino acyl tRNA into the A site. And this can actually lead to an incorporation of an amino acid at the stop codon, and then the ribosome can continue in the proper reading frame and make the rest of the full-length protein. So what we're looking for is small molecules that can reduce the fidelity of this process and push the equilibrium to this 1,000 to 1 termination to elongation ratio to maybe 1% elongation or 5% or 10%. And so we're trying to push the equilibrium over toward read-through of the premature termination codon. And if we can get up to between 1 and 10% in many diseases, this could allow you to reach a threshold for therapeutic benefit. All right, so what we did was we did a high throughput screening of over 750,000 compounds. This is done in collaboration with the Roe Lab and Southern Research. We used a Nanolic-based reporter that has a premature stop mutation, a UGA in the Nanolic gene, and then it's fused to beta globin, which has introns, which makes it susceptible to NMD. So this reporter will give us a readout of both read-through and changes in mRNA abundance. And as you can see here, the great majority of the compound screen gave us little or no increase in Nanolic activity, but there are many up here that did induce read-through. So what I'm going to talk today is about one of such compound, SRI 37240, which promotes read-through at PTCs. So we have a series of different Nanolic reporters that can read out for both read-through and NMD, and here's an example of that. This is like the reporter we use for the screen, and we see a nice dose-dependent response with an EC50 of about 6.8 micromolar for 37240. A read-through only reporter does not have the globin moiety on the reporter, so it's not subject to NMD, and that shows about a 2.9 micromolar EC50. We also have NMD-only reporters that have a wild-type Nanolic, but there are introns in the globin, so that only changes as a function of mRNA abundance. And we see a slight effect on mRNA abundance, maybe 1.5-fold. We think that's probably a secondary effect due to increased ribosome transit on the entire length of the mRNA. And then a fourth reporter just has wild-type nano look to show whether or not we're seeing PTC-dependent effects or other things such as increased promoter function of the CMV promoter, which drives our reporter. And as you can see, we don't see much of an effect there. So here's the molecule 37240, and the bottom line is it seems to have good characteristics, so we pursued this further. First of all, we did a ribosome profiling experiment. This is looking at the position of ribosomes on mRNAs. DMSO is our vehicle alone control. We use G4NA as control. It's a well-characterized small molecule that affects the accuracy of the ribosome. It alters decoding and recognition of the codon-anticodon complex. And as you might expect from making the ribosome more accessible to read through, we get a reduced peak on the stop codon. And I should point out that this is looking at a large volume of stop codons, normal stop codons in the cell. So with these normal stop codons, G4NA reduces the dwell time on the stop codon, suggesting that it's reading through, it's dwelling on these and releasing more rapidly. In contrast, 37240 has a much slower, higher peak than DMSO. When we blow up this region just past the stop codon, we see that G4NA is in fact reading through these stop codons, these natural stop codons to a certain extent. In contrast, our DMSO control and 37240 are superimposed, suggesting that 37240 does not promote the read-through of normal stop codons. So we have a small molecule that does suppress premature stop codons, but does not suppress normal stop codons. And this work was done by Jamie Wangan and Rachel Green at Johns Hopkins. All right, so then the chemistry group at SRI got involved, and they made a large number of derivatives of 37240, and one of these was named 41315. And you can see in red the changes that they made. And then we also moved to 16 HPE cells, which is a more physiologically relevant cell line. And when we did that, we found that 37240 had very limited read-through in this cell line. However, when we mixed it with a low level of G4NA, we were able to show an enhancement of read-through. Here is G4NA alone, and this is increasing amounts of 37240, so you can see it's increasing. So we're getting an amplification of the read-through effect. In contrast, 41315, which is here, 41315 alone, we're seeing significant read-through with that small molecule by itself. When we combine it with a G418, we see a further enhancement. So this is promising. It shows that this compound in the 16 HPEs is much more effective than the 37240. All right, so then the question became, does this extrapolate to the CFTR activity? So here is equivalence currents, and you can see that not much is going on with 37240 or 37240 plus G418. However, 41315 is giving us a little bit of a signal. And then when we go to G418 plus 41315, we get a stronger signal. So here's a quantitation of this data. Here is G418 alone. 37240 doesn't do much, as we saw in the reporter assay. 37240 plus G418, we see about the same as G418 alone. 41315 has a significantly higher level of CFTR activity, which is further amplified by the G418. So we're getting up to a level of about 100 AUC units, 100 AUC units, which is about 7% of wild type CFTR activity. Still a ways to go. All right, so now let's talk about mechanism for a minute. So we've looked around and we found that 37240 and 41315 specifically appear to destabilize ERF1. So this is a curve showing the abundance of ERF1 as a function of 41315 dose with a DC50 of about 1.6 micromolar. So here is an example. Here is ERF1 and ERF3, and here's the control GAPDH. When we treat cells with 41315, we lose the ERF1 band. However, when we add the proteasomal inhibitor MG132, this band is restored, indicating that this degradation is mediated by the proteasome. All right, to confirm this, we basically ask if MG132 could inhibit read-through. So here's our reporter assay. Here is a relatively modest level of 41315 when we're getting significant read-through. Here is the same level of 41315 with MG132, and you can see a significant reduction. We had to use an intermediate level of MG132 because the wild type controls were going down at higher doses. But this is clearly showing that MG132 is reducing read-through in the Allelic assay, and here's the same experiment with CFTR, and we see the same effect. So this indicates that ERF1 is being degraded in a proteasome-dependent manner to induce read-through. All right, so how is this working? We have a model based on a series of compounds called thalidomides. These compounds were shown to bind to an E3 ubiquitin ligase and actually serve as a molecular glue to recruit what are called neo-substrates to the the E3 ubiquitin ligase, which leads to its ubiquitination and subsequent proteasomal degradation. So that's our working model for what's happening with 41315 and ERF1. All right, so now let's change gears a little bit. So remember, I told you that we were getting about 7% CFTR activity with 41315 plus G418, and that's not enough. So we've decided to see if we can take different read-through molecules that have different mechanisms and see if we can enhance this stimulatory effect further. There's a number that are known. Here's our ERF1 degraders, 41315 and 41765. There's also ERF3 degraders. Adelurin affects release factor activity. There are molecules that affect tRNA modifications. The aminoglycosides affect decoding site. Translation rates are affected by Y320. We can get analogs incorporated into mRNA, such as clitazine, and then macrolides affect the peptide in the ribosomal peptide exit tunnel. So we've looked at a few of these. So what we've done here is we're giving a very low dose of G418, and then we have looked at these various compounds, and you can see most of them can enhance the activity obtained with G418. This on the right side is a series of G418 enhancers that we selected with another high throughput screen of about 500,000 compounds. And so you can see we're getting pretty robust activity. We're now bumping it up past 100% of the G418 EC50 to closer to 400% of the G418 EC50. So this is promising. We then went beyond that, and we are looking at not only our G418 enhancers here, plus the G418 EC10. We're also titrating in ERF1 degraders, ERF3 degraders, and a SMUG1 inhibitor. And you can see as we increase the number of compounds, we're getting more and more read-through. And now, instead of being at the 400% of the G418 EC50, we're climbing up to 2,000 to 3,000% of the G418 EC50. So it's clear that we can direct much more efficient read-through if we hit the ribosome, the translation termination machinery at different mechanistic levels. All right, so summary. SRI 37240 showed a strong PTC read-through and modest mRNA stabilization in FRT cells. Ribosome profiling showed that it induced a significant pause at the stop codon and showed little read-through at NTCs or normal termination codons. The derivative 413 and 5 restored a significant level of CFTR activity in 16 HBEs or immortalized human bronchial epithelial cells. 413 and 5 appears to promote read-through by enhancing the proteasomal turnover of ERF1. And our preliminary work with combinations of read-through compounds with different mechanisms shows a further significant enhancement of read-through. And we're now up to ranges with the nanolink reporters of 20 to 30-fold above the G418 EC50. And we are in preliminary stages of testing these in the Cftr assay, and preliminary results shows at least the double and triple combinations are also showing enhancements. So further studies are required to see how far we can take that. All right, these are the people that did the work. In my lab, key people were Ming Du. And in the CF Center, Steve Rowe and his group, Vinky and Jay. Southern Research, Bob Boswick did the high throughput screening, and Corinne Augelli Zafran is the lead chemist. Martin Mintz's group at the CFFT labs helped a lot. And then Rachel Green at Johns Hopkins did the ribosome profiling. Thank you very much.
Hello, everyone. I'm John Lewick, and along with Gaurav Sahai, we will be moderating this session. So the overarching theme for this session is to discuss emerging strategies being developed to restore CFTR activity when there is no protein to target by modulators. So 10% of CF causing mutations consist of frame shifting mutations, deletions, insertions, and nonsense mutations that have one thing in common, in that there is a complete or near complete loss of CFTR protein. So therefore, the therapeutic target must be shifted from protein to either the genome, transcription and translation processes, post-transcriptional processing of mRNA, or the use of total gene replacement. So I'd like to thank the speakers for sharing with us their research that is using several different approaches to achieve the same goal, which is to rescue therapeutically meaningful levels of CFTR activity when modulators have failed to do so. So at this time, I'd like to ask the speakers to introduce themselves, and then we can get on with the Q&A. So starting with you, Paul.
Hi, I'm Paul McCrae from the University of Iowa. I'm a pediatric pulmonologist with a long-standing interest in CF lung disease pathogenesis and therapies for CF lung disease.
Phil.
I'm Phil Santangelo. I'm a professor in the Department of Biomedical Engineering at Emory and Georgia Tech. My work's mostly centered on mRNA-based therapeutics for the lung and for other organs. They include both mRNA-encoded antibodies and a lot of different CRISPR constructs, most of which I didn't talk about today.
Marie?
Hi, my name is Marie Egan. I'm a pediatric pulmonologist, a professor in cellular molecular physiology and pediatrics at Yale. I have a long-standing interest in CF lung disease and new therapeutics.
David?
Yeah, I'm David Bedwell. I'm a professor and chair of the Department of Biochemistry and Molecular Genetics at UAB. I've worked on translation termination and nonsense suppression for the last couple of decades. And I think we're finally making a little bit of progress.
Gaurav, you want to introduce yourself?
Hi, thanks, John. I'm Gaurav Sahai. I'm an associate professor at Oregon State University, working in the area of drug delivery to the lung and specifically for delivery of the CF gene. So with John, again, I'd like to reiterate, we are so thankful for a great presentation by all the speakers, and we look forward for a very interesting session with this live Q&A. Back to you, John.
Yeah, so I think just to start off, maybe I'll open it up to the panel. I think, David, you hinted that, or you said that 7% isn't enough for rescue, and that you're getting there. So what are the speakers' kind of thoughts on how much CFTR activity needs to be rescued to be therapeutically relevant?
Publications have said anywhere from 5% to 30% or 35%, but I think the general wisdom is in the 30 to 35% range. Is that correct? Anybody else?
I think it probably depends on how much of a correction do you need to have an effect, a robust effect, right, or a complete correction. I know if 7% or 10% would be enough to modify the disease versus the 30% that you're seeing in your highly effective modulators.
I think the published studies that did cell mixing experiments of various kinds with, different proportions of CF and non-CF cells come up with numbers from anywhere from 5 to 50%, depending upon whether you're measuring chloride transport or bicarbonate transport using cell lines or primary cells. Certainly we know that 50%, which is what we assume heterozygotes or carriers have, is probably pretty good. We also know that there are people that have CFTR mutations that have low levels of normal transcript that have really mild phenotypes. But I think with the new tools that we're talking about, testing them in robust animal models and trying to get a handle on this at the cellular level will be important going forward.
So,
Paul, I want to ask you one thing. Sorry, if do you think it's going to be the same percentage of cells, whether are all cells equal?
That's a great question. I was thinking the same thing. What do you guys think?
Yeah, it's confusing. It gets more confusing all the time, doesn't it, with single cell RNA sequence data? It suggests that, there are cells that have low steady state levels of CFTR, like the secretory cells that people were talking about earlier today, the work from UNC, that's a really important source of CFTR. And then, of course, there are rare cells that have abundant CFTR expression, like the ionocyte. And I don't think we know right now exactly, is one more important? It seems like a lot of cells with a little might be important.
yeah, it's interesting. There's not a magic number to reach. Probably it's more of a schmear, more complicated than that. Kind of transition is there was a question for you, Paul, about if you're testing your strategies in other animals besides mice.
Well, right now we're trying to improve delivery in the cell culture model, and then we are interested in testing these in models, including mice. As Marie mentioned, the stop codon reporter mice that Craig Hodges and colleagues have developed are an attractive first place to test gene editing techniques for stop codon mutations. So we're exploring that also. And we would be interested in other models. Right now, there isn't a panoply of stop codon mutations in other animal models to explore. So we've focused on first testing these in reporter models in mice.
So-
Go ahead, John. Sorry.
So maybe as a kind of to open this up, does it seem that the models for these types of mutations need to be developed in order to kind of push this research further?
Unless you take something that has a normal CFTR and introduce a stop codon, and if you want to edit something in, right, then you could probably get some information that way. But yeah, I think that's been a hard stop to move some of this forward in animal models if you are really looking for correction in a model other than a mouse.
Yeah, I should add, I showed a pilot experiment with rhesus monkey cells. We're exploring with Alice Tarantel an experiment to look at base editor delivery to rhesus monkeys. The challenge there is that the only readout will be obtaining epithelial cells and sequencing them, which can be done, but it's nice to have a reporter along the way to give you another readout. So I think that's a challenging approach that we're exploring, but I don't have any data to report.
Just as a follow-up, Paul, there was also a question, so I'm just picking it up from our audience, like how well can you transfect ALI cells, especially also the base editors can be quite large. So there were two questions: how well just within the ALI is your transfection, and can you do how much is the carrying capacity with the peptides for these base editors?
Well, so I think, we need to explore all means of delivery technologies for base editors. A couple things that we're working on. So first is this peptide-mediated delivery. So there really isn't a limit on the cargo of these. This is a protein delivery technology where you have a non-covalently linked amphiphilic peptide in aqueous solution with a protein, and kind of akin to antimicrobial peptides that have cationic moieties. It transiently perturbs the cell membrane that allows proteins to go rapidly across the cell. So that's what we know about the mechanism. So we're exploring that in vitro and in vivo. We're also interested in viral delivery technologies because of past experience with that. And, there with AAV, for example, you do have the challenge that the coding sequence for an ABE editor, for example, is 7 kb-ish, so it doesn't fit in one AAV. So David Liu's lab has demonstrated that with dual AAV vectors with intene-extene technology, that it's possible to efficiently deliver this cargo to, for example, tissues in the CNS. So we're exploring that also. And then I think, non-viral delivery technologies are also important to look at, perhaps with mRNA cargos or with RNPs also.
Phil, do
It
you
is
want to
fairly
comment?
doable. I was just going to say it is fairly doable to make mRNAs. I mean, we've made activators and repressors and out to 7,000-8,000 nucleotides without too much difficulty. You have to know how to handle the mRNA. You need to know how to deal with it because it can hydrolyze fairly easily. But at the same time, once you kind of know how to do that, it's actually quite doable. So just a point.
So I have a follow-up on that, Phil. So I noticed in your data that there seemed to be a trend with smaller RNAs. You had higher efficiency than with larger RNAs. And so is there a cargo limit to your approaches, or does it affect it?
No, it really comes down to just dose. So we tend to use, so many mgs per kg. And so honestly, if the RNA is very long, you deliver fewer molecules. I mean, that's basically it, is what it comes down to. But I would say we haven't really seen, I mean, we've used cargoes out to 7,000-8,000 nucleotides long and been able to deliver those. So I would say I don't think that's necessarily the case. But I mean, yeah, you can deliver larger RNAs, but in general, you still have this limitation that you do at a given dose deliver fewer of them. And so multiple dosing is certainly something that can be done. And we've done some multiple dosing too. And so that's also quite feasible. We haven't had issues with increased tox or anything like that from multiple dosing.
So there's a follow-up to that, is that there are some comments about a lot of the data that you were showing is about mRNA levels that are being achieved in cells in the lung. What about protein? So nanoluciferase you showed, which is a very sensitive assay. Do you have protein results that show that you're getting a efficient expression of the protein of the mRNA you're delivering? And then also, what cell types do you think you're getting those to in the lung?
So there's a lot of other data where we've done basically a lot of, at least in terms of cell types, a lot of what we do is we combine RNA scope for tagging the RNA, so looking for the RNA, and then at the same time looking for various gene markers for the cell type. So we've been mapping that. We certainly hit some airway epithelial cells. In addition to that, we're also seeing a lot of alveolar, both type 1 and type 2 epithelial cells too. But I would say it's a combination of those two, probably slightly biased towards the alveolar epithelial cells, but also hitting airway cells. Some of that, though, is partially due to the nebulizer, to be perfectly honest, because the reality is that, the droplet sizes are actually better. They're basically better. They're sized for humans, not for mice. And so to some degree, what we see is we see the distributions are good, but at the same time, depending on the species, we definitely see some variation. And I think a lot of that has to do with deposition of those particles in different parts of the airway. So it's not necessarily the formulation that's governing that. It's really more to do with the droplet sizes themselves. And a lot of the nebulizers have been designed for really getting to the deep lung. So we're actually looking at, sculpting that droplet size distribution to make sure they get to everywhere we want them to get to. But that's a slightly different issue than the RNA and the polymers. Now, the other question you asked about is, so yeah, we do Westerns, we do, there's other quantification of protein levels. I mean, with antibody in particular, we look at how much antibody we're making. We look at function of that antibody. And from a CRISPR point of view, it really came down to, at least with Cftr, was, are we seeing a therapeutic effect? Are we knocking down RNA levels? So I guess, in terms of measuring amounts, it seems somewhat arbitrary. It really comes down to, does it do what you want it to do? Have you made enough to get the therapeutic effect you want? I mean, I would say with, at least with CF, I mean, we haven't done any CFTR studies because that's just not the space we've been working in. But I would admit that I think it's something that could be delivered probably reasonably well, actually, with our approach. I mean, one thing we have compared to some, I'll just put it, other LNPs, at least to some of the LNPs that have been used clinically, and they're nowhere near where we are. And I think it's really about screening. You just need to look at, a larger chemical space, and you might be able to find an LNP that works well too. I mean, we've done some work with James Dahlman recently showing, basically screening LNPs, but it really comes down to looking at the chemical space. And so I'm not sure who the winner will ever be, but I'm sure you can find multiple winners actually, in terms of, as long as you can screen a large enough chemical space. If you can't do that, then it's going to be tough sledding, is basically my point of view. But I would say, yes, we can get therapeutic levels of protein expression, at least for the models that we've looked at, and the distribution we hit a lot of different cell types, and I don't have that data in there, but we basically mapped them within the lung, and I just didn't show any of that because it would take too long. But we do that. We do typically look to see exactly what cell types we're hitting and how much RNA per, we quantify with RNA-scope, how much RNA are we getting per cell type. We don't do that a lot. I mean, single cell sequencing would be something else that we're looking at for the future, but right now we've been doing most of that type of work mostly in situ.
Great.
Phil, just to follow up, that was just a fantastic talk. And I think you're absolutely right. We need more materials to go to the lung. And I think polymers are working really well in that space. The questions I have is, one, if you've thought of CF would also have mucus barrier that you have to pass through. And what are your thoughts on that? And secondly, with these polymers, how polydispersed are they? Like, can they be scaled up, or they have a large polydispersity? You go for scale up, and they do not do what they're supposed to do.
Yeah, so far, at least in terms of scale up, we are working with actually a few companies to actually make much larger batches, and so far, they've been able to mimic what we've done at smaller scale in the lab without too many difficulties. So I think there's a high probability of being able to scale them up and make them, say, GMP. I think that's actually doable, though. I will admit that there are these little challenges and workup issues that you have to work out with them to make sure that you can do that. But I don't think they're certainly not insurmountable at all. I mean, a lot of the initial batches we've gotten from a few different chemical companies actually have looked pretty good. They're not quite where I would like them to be, but I'm being picky. They're pretty close, actually, and they can make them with that process at scale. Your other question had to do with, remind me. Mucus. Yeah. So right. I guess I've been kind of surprised by that. So at first, I was concerned about mucus, but there's an experiment that's not in that slide deck where we actually delivered to a cow with respiratory disease. And I know it sounds crazy that we're delivering to a cow, but there was a reason for that. It has to do with bovine RSV. But the point of the matter was that this cow actually had a fair amount of mucus in its airways, and we were still able to get a reasonable, the delivery was more efficient than I would have imagined, actually. So I'll admit, though, I have no idea how this is working. So I don't know. We're speculating about the free, because there's a certain amount of free polymer too. That seems to be important in terms of getting through mucus. It may be actually changing the charge of the mucus also, because obviously there's a lot of DNA in mucus, if I remember correctly. And so I'll admit, I don't know why it's doing this, but it appears to be getting through at least reasonable amounts. That cow actually had a fair amount of respiratory disease. So it was not CF, obviously, but it was higher levels of mucus than you have in a normal animal. And so we were a little surprised, but I have to admit, mechanism of penetration of mucus is something that we haven't gone there yet, but we know that it's something we need to look at. But I think even screening in animals with higher levels of mucus would be also interesting, because maybe some of the polymer formulations that will work better in that mucus environment versus not. There's a lot of variables, unfortunately. And so I'll admit that probably improved screening in CF models would be important, or models that you think have higher mucus, but we just haven't done that yet.
Marie, do you want to comment on that with your approach?
Yeah, I think that for us, the changing, and again, I'm the person who does our polymer discovery and optimization is really Mark Saltzman's lab. But with the polymers, we do this in our animal models. We age our mice too, which is not something most people do, because you actually get more lung disease and you get some mucus and more trouble in aged mice. And then also growing the primary murine cells at ALI on permeable supports, you can get quite a bit of mucus to build up if you age your cultures. So and then delivering directly to your cultures. We have had success with a number of polymer blends to get through mucus, which and I'm not really sure the mechanism to date yet either, but it does seem to work, but it may be charge related or
there was a question also for you, Marie, about the context in which your PNAs will work. So does it need to be a purine-rich region or?
So yeah, I think that's from Patrick, right? So I think historically it used to that was required. With some of the newer PNAs and some of the modifications, that's not essential anymore. So that actually opens up the sites that you can use much more. And then the distance from your mutation or your area of interest. I'd have to say that we've had the most success. It's something that's at somewhere between 100 to 300 base pairs in distance. When you start to get past 300, 400, 500, we see less efficient editing. Great.
So then there's been a couple questions about toxicity, and a lot of them has been directed towards your stuff, David. What are some of the toxicity with these small molecules, if any, that you're seeing?
Right. So different small molecules have different levels of toxicity. Even in the same class and mechanism, there are different levels of toxicity. So one of the things we're hoping to be able to achieve is by doing combinations with 2 or 3 compounds, we can use much lower doses of each. So for example, the combinations we did with our ERF1 degrader 41315 with G418 was using the EC10 of G418. It's only 10% of the maximal therapeutic dose. So we're hoping, and we've also been looking at, can we substitute ELX02 with G418? That's in phase 2 clinical trials for CF. And we can get a similar effect with an EC10 of ELX02. So we're trying to, work this out so that we're minimizing tox and maximizing the therapeutic benefit. But, it's still a work in progress.
So kind of a general question for the panel is that, and this is kind of to continue on with your thought there, David, is combinatorial therapies. So the idea here is to get enough protein to then maybe have a target for a modulator that exists, or even with not using modulators, what are people's thoughts on using combinatorial therapies to achieve therapeutic rescue?
I thought it was a crazy idea until Vertex came along, and now it doesn't sound so far-fetched, does it?
That's right. So does anybody else have thoughts on that? Do you think there'll be need to reach therapeutic levels with combinatorial therapy, or do you think that there will be the ability to have a one approach kind of method?
One and done.
Yeah.
I think we're going to wind up with. It would be lovely to do a one time and the single approach. I think that's quite a bit a ways off, and I wonder whether we're going to need to do something that's more nuanced prior to that achievement.
Certainly, if we had an efficient gene addition approach, that could be something where it doesn't require combination therapy. In theory, with the base editor examples, I showed all the cells were compound heterozygotes, in some case with delta F. So you could imagine if you did have an efficient base editor that could fix a stop codon mutation, you still would expect benefit from combination therapy in that setting.
So along those lines, with the gene correction, the gene therapy, it really matters which cells you're targeting. And are you hitting stem cells or just the random epithelia that's going to turn over? And both Phil and Marie were showing efficacy after a few months. You could see your, reporters. So does that mean you're already hitting stem cells, or are these cells just hanging around that long?
In our in utero work, I think we're hitting progenitor cells. I think that, and that's being delivered systemically, or those are most robust in the IV in utero experiments. In the airway epithelial cells in our mouse model, I think after 4 weeks, we need to redose. So I would say we're not hitting anything that's going to hang around.
That's a really good question. There isn't a ton of information about what the half-life of cells are in different regions of the lung in health and disease. Some of the best data are Bridget Hogan's studies. There's some mouse studies that suggest surface cells could have a half-life of over a year. So, it's a goal with a lot of these technologies is to hit progenitor cells, which vary by region in the lung. But, you have to do a long study to look at duration, I think, durability.
So a question for Paul and Marie is you show certain percentages of rescue in cell types at the genome level. When you get your technologies into the cells, do both alleles get affected? Do you think so, or is it that you'd spread over multiple cells, so there's only one allele being affected? And this would matter if you're getting, let's say, Marie, you said you had 9.2% rescue. Is that both alleles in one cell, which really actually lowers your impact over the widespread, or is it that one? So I guess the question is, if it gets into the cell, it's really active, or does it go over and spread over several cells?
When we've looked at this, just in I don't have the answer to that. I have a feeling that I don't think we hit both alleles in one cell. When we've done this, even in cell culture, and then we've diluted and cloned out single cells and tried to, and then done sequencing, Sanger sequencing, it's usually one allele that we can get corrected. I have not seen a time where we've had both alleles corrected, but again, we would have to we're looking at that much more carefully.
In the case of the adenine base editors, it depends on how many A residues are in the editing window. So if you are lucky enough, like in the case of R553X, that there's just one in the case of the mutation, then your editing would be allele specific editing. But if there are other normally expressed or occurring A's in that window, it's possible to have bystander edits that could occur on either allele.
I have a question which is sort of like, not exactly to the topic we are discussing, but I think in general it does. So, and this is a question for the panel, including John. So what do you guys think, like if you had to make a choice, you're a person who's about to give $1 billion for a clinical trial or millions of dollars, what data would you like to see? You had the best polymer or the lipid, which cell type, what are the steps you would say, check these off, and then I'll give you $100 million to do a clinical trial, and I am sure this will be successful. I'm sure this is successful is a bit high mark, but what are the key steps and the cell types, animal models you like to see, either small molecule or, like these gene editing technologies, especially gene editing technologies work, just to, open this to the panel.
Well, I could start off by saying I think it's necessary for at least for gene delivery is to have a large animal model to make sure that you can actually deliver what you think you can deliver into the areas of the lung that need to receive the therapeutic. And so
which will be this animal model? I'm just making a checklist. So this will be, what is this animal model? Ferret, pig?
What are your thoughts, Phil and Paul and Marie, on that, and David? I think pig. I'll start off by saying I think pig is a pretty good model.
I would think a pig would be a better model.
That's good.
Sheep.
Yep.
We've done pigs recently. I didn't have it in there, but we did a pig recently.
We'll throw
Well,
in a cow too. A cow would be a pretty good model.
The pig was actually better than the cow, actually, to be honest. But I think some of that had to do with technical issues. But I would say it did work well. I would say we were a little surprised, actually, but pig was okay.
Well, if you look at Vertex's work, they thought primary HBEs were all you needed. So there's the two extremes, and then you have everything in between.
Yeah, I would think that for small molecules, having a human primary cell might be a more appropriate model.
So, right, because too, David, for those clinical trials, you could give your compound to healthy controls, right? As when you're setting up a clinical trial, if you're going to deliver a nucleic acid, the FDA does not allow you to give this to like a normal, unaffected human control, right? So if you could do that to a healthy person, a small molecule, if they wanted to volunteer for such, you could see if they're going to be off-target, bad side effects, right? But you can't do that in a nucleic acid trial, at least not in this current atmosphere, I think. So you're only going to be treating patients and the most vulnerable. So I think the start for your clinical trial is very different than it is with a small molecule.
So that justifies a large animal model.
It might, and I think you'd really want to see not just delivery, and not just RNA. I think you need to see protein, and I think you need to see function, because again, when you give it to a human, this is going to be a trial. The first trials are for safety. They're not going to really be for efficacy. And those people aren't going to be able to contribute to a trial for like a year or 2 years. Is usually once they get a dose, that's it for quite a long while.
Yeah, I think you want to definitely, first of all, I won't do anything for less than 200 million. So just put me out on this thing. But I think you first want to check in a robust in vitro model, and then with success there in human cell models, if depending upon the delivery technique, animal models can get tricky. So in the case of some of the AAV capsids, for example, they have species specific tropisms that may work well on human cells, but in a given animal model they don't apply. So the only way you could test in a animal model might be to change that capsid, which would not be the tool you would use in the clinical trial. So, I mean, you definitely want to have large animal or small animal safety studies that are robust. But depending upon how you were going to deliver the reagents, there could be some species-specific differences that could lead you astray potentially. And I think you have to keep those things in mind in the design of the preclinical studies.
Any other thoughts?
I have actually kind of a specific question for Paul. Maybe you knew this one was coming, but do you know what the relevance of those bystander mutations are for the channel function?
Yeah, so with the W1282X editing that we did, there's an adjacent A at 1283, and when you edit that, it introduces a non-synonymous change. We're actually making that mutation and going to test it in FRT cells because we don't know the functional significance. There are 6 patients with a methionine at that position in CFTR2, and it's stated to be a CF mutation. So you might expect that it could be deleterious. And there's one patient in the sick kids database with the G mutation that we introduced that states patient has CF, but there's no functional information on this, so we plan to look at that. I wouldn't advocate for making bystander edits that change the amino acid sequence with base editors. And I hope that with the evolution of these tools, we can be more specific and, remove the possibility of bystander edits. So I think we're still in a, evolutionary phase of these technologies.
We published a paper a couple of years ago where we were looking at the different amino acid variants that were being inserted by nonsense suppression. And we found that some of them did have an effect, but we could rescue a significant portion of the activity, if not normal activity, with the correctors. So it's possible that you could correct with the bystander effect in some fraction of cells and still correct it and make it work.
And then maybe just for more of the gene therapy or gene delivery type approaches, have you found for all of the people doing this, is there cell types that you found that are refractory? So even after repeated delivery, there's just some cells that aren't getting your cargo.
Well, with read-through, we find that laboratory strains, we can get a ton of read-through, and the closer we get to an animal or an animal model or people, the harder it gets. And one of the things we've learned, a paper just came out that said, tumor suppressor genes and senescence, when you induce senescence, it suppresses read-through. And it turns out HEK293 cells are missing P53 and RB. And so they read through like crazy because they can't senesce, right? So that kind of stuff, basic scientists tend to use the cell lines that work the best. And that's not really what you're working with in the clinic, right?
Right.
That's the problem.
It struck me from some of the work that you did, from the results you have, Paul, is that one cell will have rescue, although it is very digital the way that you're reading this out with turning on TD tomato. If you were to then re-deliver with your base editing, would the neighboring cell also then get edited, or is it just that cell is always going to be the one that is getting your cargo?
I don't know if I really know the answer to that. Our collaborators at Feldon Therapeutics have looked at repeated delivery more than we have. And it does appear that with certain peptides, you reach a plateau of how much you can deliver. And even with repeated delivery, there may be cells that do not take up cargo. We have characterized the cell types that we can deliver protein a bit in the cell culture model and in mice, and we've seen delivery to ciliated and secretory cell types in the surface epithelium. We haven't seen delivery to basal cells with the peptide mediated delivery. We have seen delivery to small airway cells with some vectors in small airway models. And some of those cells are surface displayed basal cells. So I think, in speaking to the persistence and progenitor targeting, we have to think about the different regions of the lung and what cells are progenitors in those different regions and whether they're targetable by apical delivery, or, I'd like to hear Maria's thoughts about the systemic delivery that, she was discussing, because maybe some of those cells are more targetable from a systemic delivery rather than a luminal application.
I don't have an answer. I think that from what we see in our systemic delivery, both in the adult animal and in utero, if we then look at different cell populations, the cells that get targeted from animal to animal can vary. We as far as the percentage of different cell and cell types that we can get within, for instance, lung or an airway I don't think we have enough data to know that yet, Paul. The one thing that I will say that we often see, though, is that if we look at tissue and look at edited alleles, there are times where we can see very patchy areas of editing, so that it's not homogeneous even in a tissue or an area where you're when you think about your cell types, you'd expect perhaps all of the basal cells or all of some cell type to get edited, and we don't always see that. So I think we have to understand what triggers not only the delivery but the editing more, because that's still an area of very active investigation.
I have a question, the same thing for Phil. Do you see differences compared to like lipid nanoparticles versus polymers in the cell types that you're hitting?
Yeah, we do see some, definitely. And we also see differences from species to species that was mentioned before too. I mean, LNPs we found have been definitely more sensitive from one species to another. So if you screen one for a mouse, it doesn't necessarily mean it's going to work as well in another species. The polymers seem to be, I would say, far more species agnostic, which is why we're showing that data in the first place. But it's been that, which was a surprise, to be honest, too. But definitely there are differences in cell. There are some differences in cell types, definitely. And so we're mapping those between some of the LNPs that we've been using and some of the polymers. And so I can give you a report on that at some point. But we are trying to really understand the differences. So, the droplet size plays a role, but also it is true that the actual chemical composition of whether the polymer or LNP also seems to play a role too. But it's not. It's not obvious completely. I mean, the differences, you really have to analyze it in detail to try and understand what the biases are between one cell type and another. So, I mean, that's kind of where we are at the moment.
I was curious, Phil, if you knew anything about the mechanism of cell entry with your polymers, and then, in terms of repeated application or metabolism of the components, how that all works.
So I would say on the metabolism side, we're getting more data on exactly how each of the polymers are breaking down. So they are biodegradable. All the ones that we're using degrade, and so we are doing combinations. Well, mostly a lot of mass spec to actually look at what products are being made and over time. So that is being done. And I think that the fact that they do degrade is partially why we don't see a lot. We're not seeing any acute toxicity, I would say, across any of the species that we looked at. So that's, I guess, a positive, but that work is still ongoing to really identify all of the breakdown products, but we are doing that. Your other question was,
how do they enter the cell in the first place?
I have no idea. So that part we're in the midst of doing. So both on ALI cultures, and then even looking at our tissues at different time points to look at the pathway that the RNA is taking. So we've published a bunch of papers with a number of folks on examining endocytic, if it's endocytosis or not, and those pathways. And so we're in the midst of doing that. So, but I will admit, we don't do much of our work on cell lines in culture. We kind of abandoned that a while ago. And so, but we are looking at human ALI cultures, mouse ALI cultures, and then also the mouse tissue itself. So we're trying to go right to the tissue and examine what that pathway is. I guess I would say I don't really find many of the cell lines that we use particularly predictive of how well our delivery vehicles work, our formulations work. And so that's the only, I guess that would be my only point, but we are looking actively at those other models.
Then kind of maybe different mutation types. So David, you've looked at, I don't know if you mentioned which 16 HBE cells your small molecule was effective with, which mutation it is.
Use G542X, although we have looked at others, we just haven't done much.
And so is that kind of one of your future experiments, right? So you want to know which of these nonsense mutations this is most effective with. And what are your thoughts on that?
That we see qualitative differences between different nonsense mutations, but they all seem to respond to some extent. We haven't done the combo stuff to see if that's really the great equalizer, to make all of them more responsive. We just don't know yet.
Yeah. I think is there any questions that the speakers would like to ask each other? We'll open it up to you.
I have one. So nanoparticles, you talk about, like small mRNAs seem to work better than big RNAs. What about small molecules? It's something in the 300 to 500 Dalton range. Can you package those, or do they just leak out?
I haven't really done that much. We work mostly with nucleic acid, so I don't know. But depending on the composition, the characteristics of that small molecule, it might be possible.
Yeah. Okay. I might call you.
Asking for a friend.
Yeah.
I think that the field has worked a lot on small molecules, depending on if they're hydrophobic, what kind of molecules to use, like polymers, or hydrophilic, then liposomes. Marie, you also unmuted yourself, so please. Yep.
I would say I would agree. I think it just depends on what your cargo is and what polymer blends you might think would be helpful, right? And where your target is.
Okay.
Again, I'd like to thank all of the speakers for great talks and for taking the time to do the question and answer period. It was enriching.
Great.
Thank you for such great leadership, guys.
Thank you.
You're fabulous. Do you have any closing comments?
I do not.
We will just like to thank all of you for a wonderful session and being there. And I think we enjoyed. We also thank all the attendees for listening in and for all their questions. So I think, and thanks, John, for organizing this. And hopefully now we can open that rosé.
Thank you, everyone.
Thanks.
Right.
Thank you.